The overall goal of this procedure is to culture organotypic hippocampal slices. This is accomplished by first dissecting the hippocampus. The second step of the procedure is to slice the hippocampus. The third step of the procedure is to separate and sort the hippocampal slices. The final step of the procedure is to culture the hippocampal slices. Ultimately, results can be obtained that show healthy organotypic hippocampal slices through visualization with a dissection scope or a microscope.
Hello, my name is Ximena Opitz, and I am a research scientist in Dr. Andres Barria's lab at the University of Washington in Seattle. I will be demonstrating the procedure for organotypic hippocampal slices culture. Generally, individuals new to this method will struggle because of the need for quickly dissect and slice the hippocampi and to prevent contamination.
Sterile dissection solution and slice culture media can be prepared up to 3 weeks in advance and stored at 4 degrees Celsius, as described in the written protocol. Before hippocampal slice cultures can be prepared, it is necessary to properly sterilize both the tissue culture hood and the equipment that will be used in the dissection. First, wipe the inner surface of the tissue culture hood with 70% ethanol. Then, place the dissecting microscope and all dissection instruments inside the hood. Prepare the tissue slicer by placing a clean piece of Teflon sheet onto the stage and mounting a new blade. Set the tissue slicer inside the tissue culture hood. Using a UV light, sterilize all surfaces and equipment for 15 minutes. Turn off the light after the UV sterilization process is complete. Working in the tissue culture hood, first prepare the 6-well tissue culture plates to be used for culturing the hippocampal slices.